There is a moment every gardener knows. You walk out to the backyard on a Tuesday morning with your coffee still in your hand, and something stops you cold. The tomato plant you were nursing since March is standing at a strange angle. The leaves have gone the color of old newspaper. The stems look like they forgot which direction is up. Your first instinct is the same instinct every person gets in that moment. You think it is over. You think about the shovel. But here is what the fertilizer industry has spent 70 years making sure you never find out. Researchers at land-grant universities across the American Midwest, the same institutions that train the agronomists who built modern farming, have documented something quietly extraordinary. In the overwhelming majority of cases where a backyard plant appears to be dying, it is not dying from disease. It is not dying from pests. It is starving. Specifically, it is running out of 1 or 2 mineral compounds that the soil underneath it can no longer provide in sufficient quantity. And those exact compounds, every single one of them, can be delivered to the root zone of that plant in as little as 48 hours using items that the average American household throws in the trash before 9 in the morning. Not items from a garden center, not powders from a plastic bag with a logo on it. Items from the kitchen, items from breakfast, items from the cutting board and the coffee maker and the fruit bowl on the counter. 5 of them to be precise. And the reason most American gardeners have never heard of these 5 methods has nothing to do with whether they work. It has everything to do with the fact that they cannot be packaged, patented, or placed on a shelf at a markup of 400%. The lawn and garden industry in the United States generates over $48 billion in annual revenue. A significant portion of that number comes from selling mineral compounds back to homeowners who are already producing those same compounds in their own kitchens every single day and throwing them away. That is not an accident. That is a business model. And once you understand it, you cannot unsee it. So start here, in the most ordinary place imaginable. The kitchen counter, mid-morning, and someone peeling an onion. Most people, when they peel an onion, make the same unconscious decision that Americans have been making since supermarkets replaced home gardens as the primary food source. They pull the dry, papery outer skin away from the white bulb, drop it in the trash without a second thought, and go on with the recipe. That skin, brittle and almost weightless, looks like the most useless part of the plant. It is not. University agricultural laboratories that have analyzed the chemical composition of common kitchen discards have found that the dry outer layers of the common yellow onion contain concentrations of potassium, calcium, magnesium, and iron that rival or exceed what is found in the edible portion of the bulb itself. Beyond those minerals, the skin carries a flavonoid compound called quercetin, a molecule that has been studied extensively in human medicine for its anti-inflammatory and antioxidant properties. Inside a plant, quercetin does a different but equally important job. It functions as a kind of internal shield, protecting leaf tissue from ultraviolet stress during heat waves, slowing the advance of fungal infections through the vascular system, and stabilizing the plant's internal chemistry when temperatures swing hard in either direction. Now here is where most gardeners go wrong, and understanding this mistake is the difference between a method that transforms a plant in 5 days and a method that does nothing at all for 3 weeks. The instinct, when told that onion skins contain valuable nutrients, is to dig them directly into the soil. Bury them near the roots, let them break down, let the plant absorb what comes out. That logic is correct in principle and nearly useless in practice. Dry plant fibers decompose slowly. The minerals inside them stay locked in the cellular structure of the skin until bacteria and fungi have had enough time to fully break those cells apart. In a backyard compost pile, that process takes anywhere from 6 to 12 weeks depending on moisture and temperature. A plant that is already collapsing on a Tuesday cannot wait until August. The real method uses water. Specifically, it uses the same physical principle that makes a cup of tea work. When dry plant material is submerged in water, osmotic pressure and simple diffusion pull soluble compounds out of the tissue and into the liquid. A handful of dry onion skins placed in a quart jar with cool tap water and left at room temperature for 24 to 48 hours will, without any heat, without any grinding, without any additional chemistry, produce a liquid that darkens progressively from pale yellow to deep amber. That color change is not decorative. It is the visual signature of potassium, magnesium, calcium, and quercetin compounds leaving the skin and dissolving into the water. Once those minerals are suspended in liquid, they are already in a form the plant's root hairs can absorb on contact. There is no further breakdown required. Pour that amber liquid around the base of a tomato plant that has started yellowing, and within 3 to 4 days the response begins. The oldest yellowed leaves will not recover, but the stems will begin to thicken slightly, and the new growth emerging from the growing tips will come in a distinctly deeper shade of green. Within a week, flower production accelerates. Within 10 days, on a plant that looked finished, new fruit sets begin to appear. The quercetin effect is slower, but arguably more important over the long term. Plants, like animals, have a form of immune response. Their ability to resist fungal pathogens, bacterial infections, and stress-induced wilt depends on the availability of protective compounds in their tissue. Quercetin is one of the primary activators of that response. Gardeners who apply onion skin tea consistently through the growing season, roughly once every 10 days, report dramatically lower rates of late blight in tomatoes, powdery mildew in squash, and stem rot in pepper plants. Not because they are applying a fungicide, but because the plants themselves are fighting back harder. There is one more piece of this story that no commercial fertilizer company will ever say out loud, because it is the sentence that ends the conversation about blue crystals in plastic bags. Onion skin tea cannot be patented. The compound cannot be stabilized into a powdered form without losing its microbial value. There is no intellectual property to protect, no proprietary formula to hide behind, no reason to run a television commercial about it. The method works, the science supports it, and the cost is exactly zero. That is precisely why it disappeared from mainstream gardening advice at the same moment synthetic fertilizers became available to the American consumer market in the middle of the 20th century. But the onion skin solves only part of the problem. It feeds the plant directly through its roots, delivering minerals that are immediately bioavailable. What it does not do is address the invisible army that determines whether those minerals can actually be used effectively. Beneath every square foot of healthy garden soil, there lives a population of bacteria, fungi, protozoa, and nematodes so dense and so interconnected that scientists who study soil ecology describe it as the most complex ecosystem on the surface of the planet. When that ecosystem collapses, which it does reliably under repeated applications of synthetic fertilizer, the plant above it begins to struggle, even when mineral levels look adequate on a soil test, because it is the microbes that convert raw minerals into forms the plant can actually process. And rebuilding that ecosystem is the job of the second method, one that most American families pour down the drain every single evening without knowing what they are throwing away. Рисовая вода. Почти каждая американская семья, которая готовит рис дома, делает одно и то же движение. Промывает зерна в кастрюле, смотрит на молочно-мутную воду, которая собирается на дне, и сливает ее в раковину. Это движение, повторенное миллиарды раз на протяжении десятилетий, является одним из самых дорогостоящих случайных решений в истории американского садоводства. The water that runs off unwashed rice carries a specific and well-documented load of compounds, surface starches, B-complex vitamins including thiamine and riboflavin, trace amounts of iron, zinc, and phosphorus, and a population of wild microorganisms that cling to the grain surface during storage and handling. In its fresh state, straight from the rinsing pot, this liquid is mildly beneficial if applied directly to garden soil. The roots will absorb some of the minerals, the starches will provide a modest energy source for soil bacteria, and the result will be a small and largely unimpressive improvement. Most gardeners who try fresh rice water once and see no dramatic result never try it again. They draw the wrong conclusion from the right experiment. The experiment has a second step that changes everything. Leave that same cloudy water in a covered glass jar at room temperature for 3 to 5 days, and something begins to happen that fundamentally transforms the liquid. On the second day, a faint sour smell develops. By the third day, that smell has deepened into something recognizable, a clean, slightly tangy fermentation scent, similar to the early stages of sourdough starter or the first day of a vegetable brine. Most people's instinct at this point is to pour it out. That instinct, in this case, is exactly wrong. That sourness is the chemical signature of a microbial bloom. Lactic acid bacteria and naturally occurring wild yeasts have colonized the liquid, consuming the starches as a food source and multiplying into a dense, living culture. The science behind why this matters in a garden context was largely established by a Japanese agricultural researcher named Teruo Higa, a professor at the University of the Ryukyus in Okinawa, who published his foundational work on beneficial soil microorganisms in the 1980s. Higa demonstrated that specific communities of microbes, when introduced into depleted or damaged soil, could restore biological activity at a measurable rate within days, rather than the months or years that natural recovery would otherwise require. His concept of effective microorganisms, abbreviated as EM in the scientific literature, has since been applied on working farms in more than 150 countries and has been studied in peer-reviewed research across 4 continents. The specific microorganisms that define an effective microorganism culture are the same ones that develop spontaneously in fermented rice water. The difference is that a commercial EM product costs money, requires shipping and handling, and carries an expiration date. Fermented rice water costs nothing, develops itself without any intervention, and can be produced in a continuous cycle as long as the household keeps cooking rice. Here is what actually takes place underground in the 72 hours after fermented rice water is applied to a garden bed. The bacteria and yeasts in the liquid encounter the existing but depleted microbial community around the plant roots. The residual starches and sugars in the rice water act as an immediate food source. The soil microbiome responds by accelerating its reproductive cycle. Population counts of beneficial bacteria in treated soil have been measured in research settings at triple their pre-treatment levels within 7 days of a single application. To put that in terms that are easier to hold in the mind, 1 teaspoon of biologically active garden soil after this kind of microbial boost can contain more than 1 billion living organisms. 1 teaspoon, 1 billion organisms, a volume smaller than a sugar cube holding more living beings than exist in the entire population of North America. Why does any of that matter for a plant that is visibly struggling today? Because the microbial community in the soil is not decoration, it is infrastructure. Mycorrhizal fungi extend the effective reach of a plant's root system by factors of 10 to 100, making water and phosphorus available from distances the roots themselves could never reach. Nitrogen-fixing bacteria convert atmospheric nitrogen into ammonium compounds the plant can absorb directly. Protozoa and nematodes, when they consume bacteria, release mineral compounds in their waste that move immediately into the root zone in bioavailable form. Without those organisms, a plant sitting in soil that tests perfectly adequate on paper will still slowly starve, because the chemistry is there, but the processing machinery is gone. Fermented rice water does not feed the plant. It restocks the kitchen the plant depends on to feed itself. And that distinction is important, because it determines how and when the method should be applied. Fresh rice water applied at moments of acute plant stress produces modest results. Fermented rice water applied consistently, once per week through the growing season, beginning in spring when soil temperatures first rise above 50 degrees Fahrenheit, builds a soil ecosystem robust enough to buffer the plant through drought, heat, and pest pressure that would otherwise require repeated chemical interventions. The dilution protocol matters significantly and is the step most commonly skipped by first-time users. Fermented rice water at full concentration carries an acidity level that can irritate or damage tender root hairs, particularly in seedlings and young transplants. The correct ratio is 1 part fermented rice water to 10 parts plain water, applied as a slow pour at the base of the plant, not as a foliar spray, and not at midday when soil temperatures are highest. At the correct dilution, the liquid is gentle enough to apply to seedlings within a week of germination, powerful enough to revive an established plant showing signs of mineral lockout, and productive enough to generate visible soil health improvements within a single growing season. The third method requires a walk to the refrigerator and an honest look at what is sitting in the back of the crisper drawer. In most American households, that inspection will reveal at least one item that has crossed the line between edible and forgotten. A tomato that has gone soft, a head of romaine that has wilted to transparency, a cucumber with a bruised end and skin starting to slip. These items have a predictable destination. They go in the trash or, on a good day, into the compost bin, where they will begin a slow decomposition that will not produce usable garden input for another 4 to 6 months. For a gardener who knows what these scraps actually contain, that destination is a significant waste. The scientific framework for understanding why comes from a German chemist named Justus von Liebig, who published his landmark work on plant nutrition in 1840, fundamentally changing the way agriculture understood the relationship between soil chemistry and crop yield. Liebig's central contribution was a principle now called the law of the minimum, and it is one of the most practically useful ideas in all of gardening, yet it remains almost entirely absent from consumer-level gardening instruction. The law states that plant growth is not governed by the nutrients available in the greatest quantity. It is governed by the nutrient available in the smallest quantity. Liebig used the image of a wooden barrel built from staves of different heights. The barrel can only hold water as high as the shortest stave, regardless of how tall the others are. A tomato plant with abundant potassium, generous phosphorus, adequate calcium, and sufficient magnesium will still stop growing the moment its nitrogen supply falls below threshold. Nitrogen becomes the short stave. Everything else is irrelevant until nitrogen is addressed. And nitrogen, in the backyard gardens of the American South, Midwest, and Pacific Coast, is almost universally the first nutrient to run short. Heavy rain leaches it out of sandy soils. Dense clay soils lock it into forms the plant cannot absorb. Repeated planting of heavy feeders like tomatoes, corn, and squash in the same bed year after year depletes it faster than organic matter can replace it. The result is the slow, almost imperceptible decline that shows up first as faint yellowing on the oldest leaves, then as reduced fruit set, then as the general loss of vigor that gardeners often misdiagnose as a disease. Green kitchen scraps, particularly overripe fruits and wilted leafy greens, carry what soil scientists measure as a low carbon-to-nitrogen ratio. In practical terms, this means that relative to their weight and fiber content, they are exceptionally nitrogen-dense. A soft tomato, a wilted bunch of spinach, old cucumber skins, the outer leaves stripped from a head of cabbage. All of these items contain nitrogen in organic forms that bacteria can convert into plant-available ammonium and nitrate compounds through a process called nitrogen mineralization. When those scraps are packed into a 5-gallon bucket, submerged under water, held below the surface with a plate or a rock to prevent floating and ensure anaerobic conditions, and left to ferment for approximately 14 days, bacterial populations in the liquid reach concentrations that accelerate mineralization dramatically. The nitrogen locked in plant cell proteins breaks down. It dissolves into the water. The liquid turns dark, develops a strong and unmistakable smell, and becomes what is technically called a plant digestate, an anaerobic liquid fertilizer with a nitrogen content that, at proper dilution, delivers an immediately bioavailable dose to the root zone within hours of application. The dilution ratio for this particular liquid is more conservative than for the other methods described here, and that conservatism is not optional. 1 part scrap digestate to 30 parts clean water. The strength of the undiluted liquid is sufficient to desiccate root hairs on contact, particularly in warm, dry conditions. Applied at that dilution, poured directly onto the soil at the base of the plant in the early morning, the response is measurable and visible. Yellowed leaves do not reverse, but the new growth emerging over the following 4 to 6 days comes in 2 full shades deeper green than the tissue it replaces. Stems that had gone soft near the base firm up as cellular turgor pressure is restored. Plants that had stopped producing new flowers resume within 5 to 7 days of a single application. The mistake that burns more gardens every summer than any other single error is the decision to use this liquid as a foliar spray. The smell is intense, the color is dramatic, and the logic seems reasonable. Spray it on the leaves, let them absorb it directly, speed up the process. The result is leaf scorch that is often severe enough to defoliate a plant entirely within 48 hours. The digestate works at the soil, at the root zone, at the correct dilution. Those 3 conditions are not preferences, they are the operating parameters of the method, and departing from any one of them produces damage instead of recovery. The 4th method is where kitchen chemistry becomes something closer to alchemy, because it combines 5 separate ingredients into a single brew that addresses not one nutrient deficiency, but the full spectrum of the most common ones simultaneously. Understanding what each ingredient actually contributes and why the combination is more powerful than any single component requires spending a moment with each one before they all go into the same bucket. Banana peels are the most widely discussed of the 5, and also the most widely misused. The peel of a ripe banana contains significant concentrations of potassium, the mineral that governs a plant's ability to move water through its vascular system, regulate the opening and closing of leaf pores, and drive the biochemical processes involved in fruit development. A tomato plant deficient in potassium produces fruit that is small, watery, and prone to skin splitting as it ripens. The edges of the leaves begin to turn brown and dry, working inward from the tip. Flower drop increases. Fruit that does form does not hold color evenly. All of these symptoms respond to potassium supplementation, and banana peels deliver it in a form that, once steeped in water for several days, becomes directly absorbable. Eggshells contribute calcium in the form of calcium carbonate, the same compound that makes limestone useful as a soil amendment. Calcium is the structural mineral of plant cell walls. Without adequate calcium at the growing tips of a plant, new cells form with weakened walls that collapse under their own internal pressure. In tomatoes, this failure expresses itself as blossom end rot, the black, sunken, leathery patch that appears at the bottom of an otherwise healthy-looking fruit and ruins it before it ever fully ripens. Blossom end rot is often misdiagnosed as a watering problem, and while irregular watering can make it worse, the root cause is almost always a calcium delivery failure, meaning the calcium may be present in the soil but is not reaching the developing fruit tissue fast enough. Finely crushed eggshells steeped in slightly acidic water release calcium carbonate at a rate that the roots can process within days rather than weeks. Used coffee grounds, the kind left in the basket after brewing a morning pot, add nitrogen and improve soil physical structure. Here is the correction that matters. The widespread belief that coffee grounds acidify garden soil is based on a misunderstanding of the difference between fresh, unbrewed grounds and spent, brewed grounds. Fresh coffee is acidic, with a pH that can fall as low as 5. But the brewing process pulls most of that acidity out of the grounds and into the liquid that goes into the cup. The spent grounds left behind measure between 6.5 and 6.8 on the pH scale in university soil lab analyses, which is almost precisely the neutral range that most vegetable crops prefer. Used grounds will not meaningfully change the pH of a garden bed. What they will do is add organic matter, stimulate earthworm activity, improve drainage in clay-heavy soils, and release nitrogen over a period of several weeks as they break down. Epsom salt, which is magnesium sulfate rather than sodium chloride, contributes magnesium directly. Magnesium is the central atom of every chlorophyll molecule in every green plant on Earth. Remove magnesium from a plant's available nutrition, and chlorophyll production falls. When chlorophyll production falls, photosynthesis slows. When photosynthesis slows, everything slows. Growth, fruit set, root development, immune response. The visual symptom of magnesium deficiency is a distinctive pattern called interveinal chlorosis, where leaf tissue yellows between the veins, while the veins themselves remain green. 1 tablespoon of Epsom salt dissolved into a gallon of water and applied at the root zone can begin to reverse this pattern within 4 days. The critical caveat is that Epsom salt treats magnesium deficiency specifically. Applied to a plant that is yellowing for a different reason, such as nitrogen shortage or iron lockout, it will do nothing, and repeated applications can throw the soil's mineral balance off in ways that create new problems. The 5th ingredient is the one that makes the entire recipe work at speed, and it is the ingredient most often left out by gardeners who read about this formula online and assume the molasses is optional. Blackstrap molasses. The dark, dense, slightly bitter product of the final stage of sugarcane refining contains iron, trace minerals, and a dense concentration of simple sugars that function as a direct food source for soil microorganisms. A single tablespoon of blackstrap stirred into a gallon of the brewing mixture creates a microbial feeding event that accelerates the breakdown of every other ingredient in the bucket. Banana peel fibers that would take 3 weeks to fully release their potassium in plain water release the majority of it within 3 days when molasses is present. Eggshell calcium dissolution speeds up. Coffee ground nitrogen becomes available faster. The whole brew transforms from a slow-drip fertilizer into a fast-acting tonic. All 5 ingredients combined in approximately 1 gallon of warm water, covered loosely to allow gas to escape, and left to steep for 3 to 5 days in a warm location, produce a liquid that delivers potassium, calcium, nitrogen, magnesium, iron, and trace minerals simultaneously, along with a population of active beneficial microbes. Applied at the base of a struggling plant at a dilution of roughly 1 part brew to 5 parts water, the combined effect begins to show within 72 hours. Within a week, a plant that appeared to be dying is producing new growth, tightening its stems and resuming reproductive activity. The 5th method is the oldest one, and its history reaches back further than the United States itself. Before the year 1913, synthetic nitrogen fertilizer did not exist anywhere on Earth. Every farm, from the wheat fields of Kansas to the tobacco rows of Virginia to the market gardens on the outskirts of Philadelphia, operated on 2 inputs: animal manure and composted organic matter. The relationship between livestock and cropland was not sentimental. It was a closed biological loop. Animals ate the crops. Their waste returned to the soil. The soil fed the next crop. That loop had sustained human agriculture for 10,000 years across every inhabited continent. In 1913, a chemical plant in Oppau, Germany began the first industrial-scale production of synthetic ammonium sulfate, using the Haber-Bosch process, a method of pulling nitrogen from atmospheric air and converting it to ammonia through high-temperature, high-pressure catalytic chemistry. Fritz Haber developed the underlying chemistry. Carl Bosch engineered the industrial process. Their collaboration is credited by historians of agriculture with making it possible to feed the population growth of the 20th century. Estimates from researchers at Wageningen University in the Netherlands suggest that without synthetic nitrogen fertilizer, the planet's agricultural land could sustain fewer than half the people currently alive. That achievement is real and enormous. What is less often discussed is the trade-off embedded in it. Synthetic nitrogen fertilizer feeds the plant while bypassing the soil ecosystem entirely. Applied year after year to the same fields, it produces high yields while gradually reducing the organic matter content and microbial population of the soil. The bacteria that convert organic nitrogen die off because their food source disappears. The fungi that extend root networks retreat. The earthworm populations collapse. The soil structure, which depends on the biological activity of those organisms to maintain its physical texture, begins to degrade. Within decades, what was once productive farmland becomes chemically dependent, producing acceptable yields only because the synthetic inputs keep arriving, and declining sharply the moment they stop. For a home gardener, the old method of manure tea bypasses this trade-off entirely. Aged manure from chickens, dairy cows, or rabbits, steeped in water inside a porous fabric container for 3 to 5 days, produces a liquid that delivers nitrogen, phosphorus, potassium, and dozens of trace minerals simultaneously, in organic forms that feed the soil ecosystem rather than bypassing it. Aged chicken manure produces the most nitrogen-dense tea of the three common sources, and a properly diluted cup of chicken manure tea applied to the root zone of a tomato plant can push it back into active flowering within 5 to 7 days of a single treatment. Cow manure tea is more moderate in its nitrogen content, but exceptional for building the humus layer, the dark, spongy layer of decomposed organic matter just below the surface that functions as the soil's long-term fertility reserve. Rabbit manure is unique among common animal sources in that its nitrogen is already in a mild, partly processed form that will not burn root tissue even when applied without dilution, making it the safest option for seedlings and young transplants. The critical requirement across all three types is age. Fresh manure from any animal carries ammonia concentrations high enough to kill tender root tissue within 48 hours. It also carries weed seeds from whatever the animals were eating, and in the case of chickens and cattle, it may carry pathogenic bacteria that have no place in a vegetable garden. Manure must be aged for a minimum of 6 months, and ideally for a full year, before it goes into a tea bag. Properly aged manure has lost its sharp ammonia smell. Its texture has become uniformly crumbly, and its color has darkened to near black. Once brewed, the tea is diluted to the color of weak iced tea, roughly 1 part tea to 10 parts clean water, and applied during the coolest part of the day, either in the first hour after sunrise or in the final hour before sunset. The reason for timing is not tradition, it is nitrogen chemistry. In the heat of midday, ammonium compounds in the liquid volatilize at the soil surface, converting back to ammonia gas and escaping into the air before the roots can absorb them. In cool morning soil, the compounds stay in solution longer, move downward toward the root zone with the water, and are absorbed intact. Old-time Appalachian farmers who used this method exclusively for generations described evening application as feeding the plant while it sleeps, a description that turns out to be biologically accurate. 5 methods, 5 liquids. Each one built from something that leaves the average American home in a trash bag. Onion skin tea, delivering potassium and quercetin immune support within 48 hours. Fermented rice water, rebuilding the microbial ecosystem that makes all nutrients bioavailable. Vegetable scrap digestate, flooding the root zone with immediately usable nitrogen. The full-spectrum kitchen brew, addressing the 6 most common deficiencies in a single pour. And aged manure tea, delivering the complete nutrient profile that fed every farm on this continent before the chemistry industry arrived to sell the same elements in a bag. The principle beneath all 5 is the same one, and it is the one the industry has the clearest financial interest in obscuring. The nutrients a garden needs to thrive are already present in your home. They are generated every single day in quantities sufficient to feed a serious backyard garden through an entire growing season. The decision to discard them is not a natural one. It is a learned habit, and like all learned habits, it can be unlearned in an afternoon. The next time a plant in your garden begins to struggle, before you reach for the shovel or drive to the garden center, open the trash can and look at what is already in there. The answer to what that plant needs is very likely sitting right on top. If this changed the way you think about your garden, hit that like button and leave a comment below telling us which of these 5 methods you are trying first. Share this video with anyone in your life who has ever lost a plant they were rooting for. And subscribe, because everything we cover here is built on the same idea. The knowledge that keeps gardens alive has always been free, and it always will be.